Good morning. My name is Abdelbary Prince, Associate Professor of Biotechnology, and I'm here today to talk to you about real-time PCR. How is real-time PCR similar to traditional PCR? In both techniques, reactions are cycled in a temperature block. Every single cycle consists of three steps: denaturation, annealing, and extension. In the denaturation steps, double-stranded DNA template is separated into two single-stranded DNA molecules. This allows the primers to anneal to these two templates. And the Taq polymerase enzyme will add nucleotide, producing a new amplicon. PCR theoretically doubles the target after each cycle. How does real-time PCR work? The real-time PCR machine has a thermal block, a bright white light-emitted diode, excites the fluorescence, and filters, excitation filter and emission filter. And this will produce a signal through the complementary metal oxide semiconductor integrated circuits. We have two chemistries, the most commonly used in real-time PCR, the cybergreen 1 dye and 5' nuclease chemistry. The cybergreen 1 is a dye that binds to the minor groove double-stranded DNA. Like gel-based PCR, cybergreen assays consist of two primers, forward and reverse primer, without probe. The primer anneals to the complementary sequence of the single-stranded DNA template, and the Taq polymerase produce double-stranded DNA. This will allow the cybergreen 1 dye to intercalate in the minor groove of the double-stranded DNA and will be excited, producing emission energy. This emission energy will be calculated and quantified. The problem with cybergreen 1 dye is it binds non-specifically to any double-stranded DNA, and the signal from non-specific product give rise incorrect quantitative result. If the primers are not well designed and the reaction is not optimized, cybergreen assay where results will include signals from all PCR products, target and non-target. The non-target PCR amplification is a commonly observed in gel-based PCR, and the gel electrophoresis separates non-target products from target if there is a size differences. There is no opportunity in real-time PCR to separate non-target from the target amplification. Additional data are required to determine whether cybergreen assay results are derived solely from target or not. The additional data are required, including sequencing the assay products, gel analysis, and dissociation curve analysis. The another technology is dependent on 5' nuclease assay, which uses a gene-specific Taqman probe. The probe has a reporter dye and a quencher. The reporter on the 5' end and a quencher on the 3' end, which also blocks extension. The 5' nuclease assay gets its name from the 5' nuclease activity of the Taq DNA polymerase enzyme. The nuclease domain can degrade DNA bound downstream of DNA synthesis. In the 5' nuclease assay, the excitation light hits the reported fluorescent dye, and this will generate energies that are absorbed by the quencher, what's so-called fluorescent resonance energy transfer, FRET. In the intact probes, there is no generation of signal, no emission, and nothing is reported. While when we have 5' nuclease activity, due to the probe is annealed to the specific sequence, this will separate the reporter from the quencher, and will generate signal, and this emission will be reported and quantified. In the PCR annealing step, the primers and the probe are annealed to the template. And following probe cleavage, the reporter is permanently dequenched. This is leading to increasing the signal and calculated the amount of PCR product, which is theoretically doubled. The level of fluorescence has increased in a permanent manner by a proportional amount. There is no mismatch extension for Tachman probes. The destabilizing effect of probe mismatches cannot be mitigated by Tach polymerase enzyme because extension is blocked by the quencher. And the Tach polymerase displays the probe rather than cleaving it. The probe returns to the quenched configuration, and there is no signal is produced from the homologue. The non-specific product exclusion is the most important advantage of 5' nuclease activity using Tachman technology. The non-specific PCR product like primer dimers, amplified products unrelated to the target, they are often formed during primers bind to the other template sequences in the same and by chance are able to form a PCR product. The Tachman probes cannot bind to nor be cleaved by any specific product. So all such products are automatically excluded from the assay results. Now we will review the different two chemistry behind real-time PCRs. They are the most commonly used now for the real-time technology. The cybergreen assay uses a double-stranded binding dye to detect accumulating PCR product. Only primers are needed, and this gives advantage of inexpensive technology. But we don't have the ability for multiplexing of different target to be determined simultaneously. On the other hand, 5' nuclease activity uses a target-specific fluorogenic probe to detect accumulating PCR product, and the assay consists of forward and reverse primers, as well as probe. The probe is expensive, and this is one of the disadvantages of the 5' nuclease technology. But this gives us another advantage, the multiplexing properties of the 5' nuclease assay. And this helps us to quantify different genes simultaneously in single tube, single reaction. Thank you for watching this video.